Okay, so welcome to this next video in which we're discussing the defects in excitation contraction coupling within hypertrophied cardiomyocytes. Okay, so we've now talked about the fact that the calcium tide, the size of the calcium tides in hypertrophied cardiomyocytes is reduced compared to the unhypertrophied cardiomyocytes. Okay, so every time you stimulate a cardiomyocyte to contract, every time you activate an action potential along its membrane, what happens is that this triggers a calcium signal which is global within the cytoplasm of the cardiomyocyte, i.e. calcium goes up everywhere. And the size of this global calcium spike in hypertrophied cardiomyocytes is reduced compared to in non-hypertrophied cardiomyocytes. And this is potentially, or at least we think is most likely, the reason that the cardiomyocytes have a lower ability to contract, so they contract with a lower force. And the reason is that if the calcium tides are smaller, then you'll recruit fewer sarcomeres, you'll activate fewer sarcomeres to actually contract. And that means that the force that the contraction of the cell will produce is lower because there are fewer sarcomeres actually generating the force. Okay, right. So we've seen that there are three things which are all contributing to that. So firstly, the dyadic cleft size is greater. Secondly, the T-tubules have atrophied. Okay, and thirdly, the circuit 2 pump isn't functioning properly. Now, the circuit 2 pump not functioning properly leads to calcium being too low in the SR lumen. Okay, so that means that when you open the type 2 ranitidine receptors, less calcium is going to come out, so the calcium sparks are going to be lower, and since the calcium sparks are what make up the calcium tide, then that means the calcium tide is going to be smaller. Atrophy of the T-tubules means that there are loads of these processes of the SR which just aren't in a functional calcium synapse anymore, and therefore these type 2 ranitidine receptors just cannot be activated to open, so they're effectively wasted. So the atrophy of the T-tubules means that the number of calcium sparks you're going to get is going to be lower, so that again is going to reduce the size of the calcium tide. Increased dyadic cleft size means that less calcium will make it from the calcium sparklet to the type 2 ranitidine receptors on the SR membrane. So the number of type 2 ranitidine receptors on the SR membrane that you'll actually activate to open will be lower, so the amount of calcium you'll get released is lower, causing the calcium sparks to be smaller. Okay, what we now want to look at is why in a hypertrophied cardiomyocyte, which isn't at rest, okay, so there's no action potential occurring, so there's no calcium sparklets occurring through these L-type voltage-gated calcium channels, so there shouldn't be any activation of the type 2 ranitidine receptors. But in hypertrophied cardiomyocytes, what you see is calcium sparks occurring sporadically throughout the cytoplasm of the cell. So type 2 ranitidine receptors are opening sporadically, basically, and we want to understand what is actually causing that. So basically, in hypertrophied cardiomyocytes, what you appear to get is overactivity of the type 2 ranitidine receptors. They appear to be more sensitive to calcium, basically. So let's now discuss this. So I'm going to discuss the structure of the signaling complex which results in the phosphorylation of the type 2 ranitidine receptors. Because basically, what's going to happen to cause this overactivity of the type 2 ranitidine receptors, which causes them to sporadically open without a calcium sparklet, is going to be that they are over-phosphorylated by protein kinase A. So I want to show you the complex involved in this phosphorylation of the type 2 ranitidine receptors. So basically, if this is the plasma membrane here, okay, then there is a protein in the plasma membrane known as adenylyl cyclase 5. Okay, so let me draw this in here. So this is adenylyl cyclase 5 here. Right, so just to discuss the adenylyl cyclase 5 structure briefly, adenylyl cyclase 5 has these two transmembrane regions consisting of 6 membrane-spanning alpha helices. So this is the transmembrane region 1, and this is the transmembrane region 2, and they both consist of these 6 membrane-spanning alpha helices. This loop between the transmembrane domain 1 and the transmembrane domain 2 is known as the C1 loop, and it can be divided into two portions. The C1a portion, which is the bit closer to the transmembrane region 1, okay, and then the C1b portion, which is closer to TM2. Then the carboxyl terminus of the enzyme over here, this is known as the C2 domain. So here's the C terminus of the polypeptide, and here's the amino terminus of the polypeptide. This portion of the polypeptide that is close to the C terminus, this is called C2, and this can be divided into two portions, C2a and C2b. So C2a is the portion closer to the transmembrane region 2. So I'll highlight this in red. So C1a and C2a are very important regions because those are the portions which will dimerize together in order to make the active adenylyl cyclase enzyme. So this is a adenylyl cyclase enzyme. So adenylyl cyclase, and it's specifically adenylyl cyclase 5 that's going to be involved in our complex. So this protein binds to another protein. So let me show this next protein that it's going to bind to. So I'll just denote this next protein as a great big blob, which seems very blunt when this protein was drawn out with such intricate details. Okay, so this next protein is something known as MA-cap beta. Okay, so I'll tell you what that stands for in a moment. Right, so this is the protein called MA-cap beta, and MA-cap beta stands for muscle-specific A kinase anchoring protein beta. So muscle-specific A kinase anchoring protein, and A kinase is just shorthand for protein kinase A, and this is the A kinase anchoring protein beta. So that's where the A cap comes from. It stands for A kinase anchoring protein. The little m stands for muscle specific, and then finally you just need this beta. Okay, right. So what am I actually talking about? Well, basically there is a complex which is involved in keeping a type 2 protein kinase A enzyme close to the type 2 ryanodine receptor at all times. So there is this complex between proteins in the plasma membrane and proteins in the sarcoplasmic reticulum membrane in the heart. So this is the plasma membrane here. This is the sarcoplasmic reticulum membrane down here. So basically now, in the sarcoplasmic reticulum membrane, we then have the type 2 ryanodine receptor sitting here. Oh, and I should draw the type 2 ryanodine receptor with a bit more detail now. Right, how am I going to do this now that I've drawn this crude picture? So I'm going to have to discuss with you a little bit more about the structure of the type 2 ryanodine receptor, because when we come to talk about why it is sporadically opening, we need to actually know something about its structure. So to draw this type 2 ryanodine receptor in a bit more detail than I've ever drawn it before, here we have it. Okay, so it's a massive, an absolutely massive great protein. In fact, it's not just one protein, it's a tetramer basically. It's made up of 4 proteins all stuck together. So here I've divided it into 4 quarters. So each one of these subunits that makes up the type 2 ryanodine receptor tetramer is a type 2 ryanodine protein. So basically it's a homotetramer. All the proteins in these 4 slots here are identical proteins. So they're all type 2 ryanodine proteins. Okay, so you take 4 type 2 ryanodine proteins, stick them together, and that will make you the type 2 ryanodine receptor. Now, a fun fact about the proteins that make up this tetramer, they are absolutely massive. They're around 5,000 amino acids in length. Now, that's not trivial. That is a massive great protein. So these things are so big that you can actually see them with electron microscopy very easily. They are massive great proteins. Okay, so this is what we crudely got shown here, just as a little channel. You can think of this basically as the thing cut down the middle. Okay? Right. Now, the important thing is that these type 2 ryanodine receptors are attached to the muscle-specific A kinase anchoring protein beta. Okay? And the muscle-specific A kinase anchoring protein beta is then also attached specifically to this adenylyl cyclase 5 protein within the plasma membrane. So it forms this sort of link between the SR membrane and the plasma membrane. Now, also attached to this complex here is a protein kinase A enzyme, and I need to discuss something about the structure of protein kinase A with you then. So let me describe to you the structure of protein kinase A enzyme. So protein kinase A is not one protein. It's made up of a complex of many different proteins, okay? So here are 2 of these proteins now shown in a dimer. 1, 2. Now these 2 proteins are supposed to be identical. I know I had to draw this one sort of squashed up, but that's simply because I'm running out of space down here. So these 2 proteins are identical, and those are what are known as the regulatory subunits of protein kinase A. Okay, and this type of protein kinase A enzyme that I'm drawing here is what's known as a type 2 protein kinase A. And basically, the type of protein kinase A that you are is determined by which regulatory subunits you use. So basically, there are 2 genes that code for regulatory subunits of protein kinase A. The type 1 regulatory subunit and the type 2 regulatory subunit of protein kinase A. These are going to be the type 2 regulatory subunit. Now you might ask, what is the difference between the type 1 and the type 2 regulatory subunits of protein kinase A? Well, basically, the type 2 regulatory subunits of protein kinase A bind to ACAP proteins, such as the muscle-specific ACAP beta, okay? Whereas the type 1 regulatory subunits do not bind to ACAP proteins. So this is a type 2 one because I've very blatantly shown it attached to the MACAP beta protein. Okay, so these type 2 regulatory subunits of protein kinase A, they are attached to ACAPs basically, whereas the type 1s are not, and they will stay free in the cytoplasm. They won't bind to ACAPs. Okay, right. So then, this isn't the complete story. You also have 2 catalytic subunits of protein kinase A bound to the 2 regulatory subunits. So we have 2 regulatory subunits in red and 2 catalytic subunits of protein kinase A, which are these little blobs that I've labelled C in here. So these are in blue. So these are the catalytic subunits of protein kinase A. Okay, catalytic subunits of protein kinase A. Okay, so you have this complex within the heart. Now, these protein kinase A complexes here, they are known as R2C2 complexes. And the reason for that is that you have 2 regulatory subunits complexed together and 2 catalytic subunits complexed together. Now, when the catalytic subunits of protein kinase A are bound to the regulatory subunit like this, they are inactive. And by the way, I should have stated this, the catalytic subunits are the portions which are actually capable of adding phosphate groups onto proteins. So they're what you may have always thought of as protein kinase A. They are the portions that are actually going to put phosphate groups onto proteins, and both of them are functional. Okay, and also one other thing to say is that the difference between the 2 types of protein kinase A is in the regulatory subunits, not the catalytic subunits. So there is only one type of catalytic subunit. This is a type 2 protein kinase A enzyme. The entire thing is protein kinase A, not just this catalytic subunits. The entire R2C2 complex is protein kinase A. This is a type 2 protein kinase A because it has the type 2 regulatory subunits and is therefore bound to an A cap. Whereas the type 1 protein kinase A looks exactly like this again, but it's got the type 1 regulatory subunits, which I would draw exactly the same as I've drawn here, except that I would not draw it bound to an A cap. Okay, now, when the catalytic subunits are bound to the regulatory subunits like this, they are inactive. However, you'll notice these little furrows that are drawn on the regulatory subunits. Those are cyclic AMP binding sites. When cyclic AMP binds in those 4 binding sites, the regulatory subunits will change conformation and release the catalytic subunits, and they will then be able to phosphorylate proteins, such as the type 2 ryanodine receptor. Okay, right. Now, this complex exists within the heart. You have adenylyl cyclase 5 enzymes linked to a MA cap beta protein, which is then also bound to the type 2 ryanodine receptor and to protein kinase A. And this is the complex that is involved in phosphorylating the type 2 ryanodine receptor in response to beta 1 stimulation. So to complete the picture, you then also need to add in, maybe somewhere here, the beta 1 adrenergic receptor, which is a G protein coupled receptor. So here is the beta 1 adrenergic receptor. And then sitting in between, you would then have the GS heterotrimeric G protein. Okay, so this now is the full signaling cascade all here. So let me just highlight these things in color to make them more obvious. So here is the beta 1 adrenergic receptor. And what will happen is the noradrenaline will bind to the beta 1 adrenergic receptor when it's released onto the cardiomyocyte by the cardiac nerves. It will then activate the heterotrimeric G protein, specifically the alpha S subunit of this heterotrimeric G protein, which I'll highlight in blue there. It will break off the GDP that's usually bound to this alpha S subunit here, and it will replace it with GTP. And then what will happen is the alpha S GTP subunit will break off from the beta and the gamma subunit, and it will come and activate the adenylyl cyclase 5 enzyme. And the way it will do this is it will bind to both the C1 domain and the C2 domain of the adenylyl cyclase 5 enzyme and promote the dimerization of the C1a portion with the C2a to produce the active enzyme. This enzyme will now produce cyclic AMP, which will go down and bind to the cyclic AMP binding sites of the type 2 protein kinase A enzyme, and then the catalytic subunits will be released onto the type 2 ryanodine receptor, and they will phosphorylate the type 2 ryanodine receptor. Okay, so this is the normal pathway. Now, in hypertrophied cardiomyocytes, what you find is that this type 2 ryanodine receptor is over-phosphorylated. So it is likely that this pathway is overactive and the catalytic subunits are over-phosphorylating this type 2 ryanodine receptor, potentially in the, that the catalytic subunits are over-phosphorylating the type 2 ryanodine receptor. And we'll see what the effects of over-phosphorylating the type 2 ryanodine receptor are in the next video, and how this is going to lead to these sporadic calcium sparks.